And it's part of the process in our bodies that makes proteins. So what you're seeing here is called the central dogma of molecular biology. And it tells us that we start with the blueprint information, our DNA, which is permanently stored in each one of our cells. And this DNA is converted inside of our cells into messenger RNA. And you can think of messenger RNA as a little bit of a middleman, relaying the information that the DNA is giving us and using it to create proteins. So proteins are the doers in our body. They're responsible for nearly all functioning within our bodies. And so really any kind of disease or condition, it almost always has some critical protein involved. Either there's too much of a protein, too little of a protein. Sometimes a protein has been mutated and so it doesn't have the correct shape or form or function. So we're excited about messenger RNA as a type of therapeutic because it's upstream of this protein production. And because it's a middleman, because it's temporary, we can actually try to fool around with its expression so that we can make changes in protein without changing our DNA. Because it's temporary, we can actually try to fool around with its expression so that we can make changes in protein without changing our DNA, without permanently altering anything in our DNA. So one of the ways that we can do this, if we want, for example, more of this blue protein here, we can simply add more of this same messenger RNA. So the messenger RNA has a particular code. We can add more of it into our bodies, and so we can augment the expression of that particular protein. Similarly, if we want to make some other protein, perhaps even a protein that our body doesn't even normally know how to make, we can deliver a different sequence of messenger RNA into our body and ask our bodies to make that protein as well. And so in this way, we can induce all sorts of protein expression that can help either treat or, in the case of these vaccines, to prevent disease. So just a little bit on what mRNA drugs can do. So vaccines are clearly at the forefront of applications that are being used in the clinic. I've been working in the area of RNA delivery for over 15 years now, and as with many of my colleagues, we've long been believers of the enormous potential of these genetic medicines. And let me just explain to you a few of the different types of conditions that can be treated with messenger RNA. So one of the most obvious types of applications is protein replacement therapy. So for example, those with hemophilia, this is a blood clotting disorder. And the reason people bleed is because they are missing a particular protein in our blood that helps the blood clot. And so a fairly straightforward application of messenger RNA would be to get that messenger RNA sequence for that missing protein, to deliver it into the body, and then our body would be able to make that protein and the blood is able to clot. Okay, so there are numerous different conditions where we need to make more of a protein or to introduce a missing protein into our system. Vaccines, as I mentioned, have been the most clinically advanced application. And in this case, we're not replacing any kind of missing protein, but instead we're introducing a type of protein into the body that our bodies have never seen before, a foreign protein called an antigen. So in this case, for SARS-CoV-2, we're introducing a small protein found on the viral surface. And when we show that to our immune system, after the cells create that small antigen, the immune system reacts in a way that it will help us respond to any kind of infection that might take place. And so to prevent that infection or to prevent a very severe response to that virus. I won't go into them in a lot of detail, but messenger RNA similarly can be used for cancer immunotherapy applications. So we can deliver messenger RNA that encodes for proteins that will help teach our immune system how to attack the cancer cells in our bodies. We can also use messenger RNA to create what are called transcription factors. These are a type of protein that help the stem cells in our body decide which other types of cells they're going to become. So we can help our stem cells create new types of tissues using this technology as well. And then finally, for those of you who are aware of gene editing or interested in it, we can actually turn down protein expression. All these other ones involve creating more protein expression, but we can turn it down by delivering messenger RNA encoding for a nuclease called Cas9 or any of the other nucleases, which can then go into our nuclei and carry out gene editing to silence particular proteins in our system. And if any of you are interested in learning more about details of these applications or to learn more about the delivery vehicles that are being used, you can reference this review paper that a member of my group wrote a few years ago. So the problem with messenger RNA as a therapeutic, we can't just take a pill, we can't just inject it into our systems and hope for the best. This is because it's a very large molecule and it's negatively charged. Our cell membranes are also negatively charged. So when those two negative charges come up against each other, the mRNA is repelled and it's not able to enter the cell. If we just inject it into our bloodstream, it will be cleared very rapidly through our kidneys. And so what we really need is some kind of a delivery vehicle that's able to package up this messenger RNA and then take it to where it needs to go inside the body, into the cells where the protein expression is needed. So I wanted to just put together a slide here that would highlight all the different things that we need this delivery vehicle to do. It's really, in my opinion, pretty miraculous that we can come up with something that works. So in the case of vaccination, we are going to receive an injection of these lipid nanoparticles into our muscle tissue. Okay, so those lipid nanoparticles then need to diffuse to the site of the cells that are going to be vaccinated. Sometimes these cells are going to be the muscle cells, sometimes they might be the immune cells, because there are also immune cells that reside in our muscle tissue. One way or another, these lipid nanoparticles will make their way to the vicinity of these cells, and they're taken into the cell through a process called endocytosis. So basically, the cell membrane reaches up and around the nanoparticle and pulls it inside. Normally, the cell does this to try to sample its environment and to find nutrients and other molecules that may be out there that it wants. When it brings in a lipid nanoparticle, it doesn't know what to make of it. It's in this walled-off compartment called the endosome. And the cell keeps it there because if anything damaging is brought in, it wants to be able to get rid of it. So the problem that these delivery vehicles face is that they don't readily exit this endosome. So the chemistry of these particles has been designed very carefully over the years to enable this endosomal escape process, as we call it, which will then allow the lipid nanoparticle to deliver the RNA into what's called the cytoplasm of the cell, which is the watery area outside the nucleus. And that is where the proteins can then be made. So really, we're asking our nanoparticles to do quite a lot, overcome a number of different barriers. And it's really decades of research that has enabled us to get to where we are today and to the very rapid development of these vaccines. So cationic lipids have been used for the earliest forms of nucleic acid delivery. So DNA is where researchers really started in this field. And DNA, although it's structurally somewhat different than RNA, it's still made up of very negative nucleobases or nucleosides. And it also, it's a very large molecule. And so some of the properties that we need to encapsulate DNA are very similar. So what researchers decades ago found was that they could use cationic lipids, so these lipids that have a positive charge, and then here is something like a 16 carbon long tail, 2 of them. And if you're able to use these these lipids to create liposomal structures that encapsulate the DNA. So here's what a liposome, a traditional liposome looks like. This is a cross section right here, and here's what it kind of looks like in 3D. So it's similar to our cells. It has a lipid bilayer all around the exterior of the liposome. And these blue balls here represent that cationic head group on the lipid, which is water-loving. So this water-loving group faces outward into an aqueous solution. And then we also have these water-loving materials facing inward. Meanwhile, the tails all come together because they are hydrophobic and they want to hang out with each other. So inside, the DNA, which is water-loving, can be encapsulated. Over the years, the technology and the exact chemistry used for nucleic acid delivery has evolved. And DNA, as I mentioned, it is a little bit different than RNA. And so as we've moved into RNA delivery, we've also had some developments in the type of particles that are used for efficient RNA delivery. So we're now at a point where these SARS-CoV-2 vaccines, they're making use of more recent forms of lipid nanoparticles. These are a bit more of what I'd call a solid lipid nanoparticle, and they're made up of several ingredients. So one is called an ionizable lipid. So it's like that cationic lipid I showed you before. However, it only takes on positive charge inside the cell. And because it's neutral in the bloodstream, it actually causes less toxicity than some of the earlier versions of these lipids. There's also a fair amount of cholesterol in these particles, just the same way that there's a lot of cholesterol in the cell membranes inside our body. So it serves a similar purpose where it helps to fluidize our particle to provide some support and shape to the particle and stability. And then there are what we call helper lipids. These are phospholipids, also can be naturally occurring lipids that we incorporate into these particles that can help with the packing. So you can see all these different molecules are packed inside this particle. This is just my kind of interpretation for what might be going on here inside the particle. These are very small particles, and so to my knowledge, you know, we only kind of have clues and bits and pieces as to what these look like on the inside. But the specific helper lipid is chosen to help this messenger RNA shown in black to really pack in there effectively. There's a molecule called polyethylene glycol, or PEG. This is a polymer that is currently found in some FDA-approved medicines, and this hangs out on the exterior of the particle. It also helps with the stability of the particles, and for other applications, it can help shield from unwanted immune clearance, although that's not as much of a concern in the case of vaccines. So this is what these particles look like. My lab works with these particles. Iju's lab works with these particles, many different types of very similar looking particles. They're all very similar. One thing that tends to change is the ionizable lipid. So this is where a lot of the intellectual property exists within the lipid nanoparticle space. And it is the major difference between the Moderna and the Pfizer BioNTech vaccines. Okay, so here are these ingredients again. Both of the vaccines use the same set of 3 other lipid nanoparticle ingredients. They all use cholesterol, they all use a helper lipid called DSPC, and they all use a very specific version of this PEG, okay? Their messenger RNA sequences are a little bit different, but essentially are going to do very similar things once they get into the body. As for the ionizable lipids, this is what BioNTech Pfizer's ionizable lipid looks like. So you can see here, it's a neutral molecule. This is what you know, the muscle tissue sees until these molecules make it inside of the cell. And this amine group right here is able to take on a positive charge that helps our nanoparticle escape that endosome, okay? So it's very interesting that if we look at the Moderna lipid, this is a very, very similar looking lipid, right? So we have an alcohol group. The distance between the alcohol and the amine is a little bit different. It's 2 carbons over here versus 4 carbons over here. And then we have similar tail structures. All of the tails incorporate this group right here. This is called an ester, and this ester group is degradable under the conditions inside of our body. So this helps to make sure that after our lipid nanoparticles have done their job, they're able to break down and to degrade inside of our body so that they can be cleared and they don't accumulate to cause toxicity. So all of the tails have this. And then one of the other differences here is that we have essentially 4 short tails at the end of the BioNTech Pfizer ionizable lipid, and we have 3 tails over on this end. So structurally very similar lipid nanoparticles. So I mentioned before that RNA is negatively charged, and these ionizable lipids, they become charged only under reduced pH conditions. So we put these materials into slightly acidic solutions in order to form the nanoparticles. So these electrostatic interactions help draw our materials together. And you can make the nanoparticles either by hand using pipettes, or you can make them in a microfluidic device. There are a number of different ways, but essentially you're going to be mixing an RNA stream, which is an aqueous stream in water, and you have a lipid mixture, which is dissolved in ethanol. And when you rapidly mix these 2 streams together, you get lipid nanoparticles, which precipitate out of solution. They tend to be about 80 nanometers in diameter, but that can vary depending on the exact ingredients that you put into that nanoparticle. So just a couple more slides from me explaining how these messenger RNA vaccines are helping protect specifically against SARS-CoV-2. So this is a depiction of SARS-CoV-2, the virus. And on the surface of the virus are what are called the spike proteins. You can see these kind of look like a crown or a corona, so that's why they're called coronaviruses. And this is how the virus, the spike protein, is how the virus enters our cells. So this spike protein can bind to another protein, a receptor in our bodies, and there's a lot of it in our lungs, which is why we tend to get sick in our lungs. So this receptor can bind to the spike protein, and the spike protein can then bring the whole virus into the cell and to cause an infection. So that's how the virus is causing infection. But similarly, when our immune system responds to this virus, you can see the spike proteins are right here on the surface. And so of all the different proteins that are present in the virus, it would be most easy for our immune system to detect these spike proteins because they're on the outside. So for that reason, that's one of the reasons why the messenger RNA that's being delivered through the vaccine, it encodes for this spike protein. So I'm going to just show you, if we have any immunologists in the crowd, this is a somewhat simplified version of how our immune system is responding to these particles. For the rest of you, it will probably already seem overly complicated, but the immune system is absolutely amazing in how it works. So if people like me, the drug delivery scientists, have done their jobs in creating a good particle that's able to package up our RNA and to deliver it into cells, here's it outside of one of the cells that's going to be vaccinated in our muscle tissue. And this vaccinated cell, again, it can be a muscle cell, it can be an immune cell, or other cells that are present in the area. So this particle needs to enter our cell that's going to be vaccinated. And again, it's in that endosome. So the particle needs to escape the endosome and deliver the messenger RNA that encodes for the spike protein into this watery part of the cell. And there it can be turned into protein by the cell. So here we have the spike protein that's being made and smaller pieces. So sometimes the spike protein can get broken down into smaller pieces. All these things are called antigens. So an antigen is this foreign bit of protein that our immune system will respond to. So some of the spike protein, the cell knows it's foreign, and so it holds it out. It holds it outside its cell, and it says, hey, immune system, please come and do something about this. So there are a couple of different ways that our immune system can respond. One way is that our B cells, there will be just a couple B cells in our body that have antibodies that are specific for this particular spike protein. It's amazing how many different types of B cells we have in our body. So when a B cell finds this infected cell, or sorry, this vaccinated cell, and binds to it, it is able to produce antibodies against that particular antigen. Okay, so in the event that an infection ever tries to take hold, so in the event that this virus, the SARS-CoV-2 virus, enters our bodies, our immune system will be prepared. These antibodies will have been made, and they can quickly surround this virus, bind to the different spike proteins, and when it completely coats the whole virus in antibodies, that marks this virus for clearance by the immune system. So we say it's been neutralized. All right, so that's one way this works. The vaccine teaches our B cells to make antibodies, which can then try to get rid of the virus before it ever even infects any of our cells. The other thing that can happen is either the B cell can engage in a signaling process, or there are cells called antigen presenting cells. These are immune cells that can help kick off this other part of the process. So a B cell can talk to a T cell called a helper T cell, which helps get this signaling process going. It will then talk to an antigen presenting cell. Differently or separately, the antigen presenting cell can detect some of this foreign protein on the surface of this vaccinated cell and gobble it up. One way or another, the antigen presenting cell can talk to a different type of T cell called a cytotoxic T cell. Okay, so this T cell can kill other cells. And the cytotoxic T cell is equipped now with this very tiny piece of antigen that it can use to try to match up with any cell that's been infected. So in the event some virus does get into our system, and in the event that a cell becomes infected, that infected cell will actually show pieces of the antigen on its surface, and it will match up with our cytotoxic T cell such that this cell is then killed. Okay, so there are a couple of different ways that the vaccine works, but the main takeaway here is that our B cells will make antibodies to try to sequester the virus before it infects our cells. But if that infection happens, we have this other part of the immune system over here that will make sure those cells are killed. Okay, so that's what I have, and I'm going to turn it over to Professor Dong.
Okay, issues. You should be presenter now. Yeah, great.
Yeah, could you see my slides?
Yes.
Let me share the screen.
Yes.
Okay. Okay, is that good? Wonderful. So hello everyone. Also good morning, good afternoon, good evening, depending on where you are. First, I would like to thank Christine, Cara, and Fabio for their great efforts on hosting this very exciting webinar. It's really my great pleasure to share with you some examples of lipid nanoparticles for mRNA vaccines and mRNA therapeutics based on our research experience. Thanks, Katie, for the wonderful presentation about mRNA vaccines against SARS-CoV-2. I don't need to give additional background information on that. In our study, we learned that both mRNA sequences and the delivery materials are critical for an effective mRNA vaccine. On the left, here's a process for the optimization of mRNA sequences. We know the untranslated regions, UTR of mRNA, play important roles in regulating mRNA functions. So we investigated 5' and 3' UTR of mRNA through two approaches. One is bioinformatics analysis of endogenous gene expression. We analyzed over 4,000 different genes reported in the literature. And the other approach is de novo design, which is based on several predetermined criteria.